This is episode 63 of the Immunology Podcast, T-Cell Tolerance with Dr. Alice Long. Hey everyone, this is Dr. Jason Goldsmith and Dr. Brenda Rout. Welcome back to the Immunology Podcast. We have conversations with immunologists. The Immunology Podcast is brought to you by Stem Cell Technologies, a global biotechnology company supporting life science research and fostering communication and collaboration in science. If you enjoy the Immunology Podcast, please rate us and leave us a review. We're always looking for feedback on how the podcast can be improved and for suggestions on guests. Today, we have Dr. Alice Long from the Benaroya Research Institute on the podcast to talk about her research on the mechanisms of immune tolerance. We also got our usual roundup of recent highlights of the immunology news coming up. But first,
IUIS 2023 is fast approaching. We are very excited to be attending as media partners. And don't forget that the Congress will take place in Cape Town, South Africa from November 27 to December 2nd. Visit IUIS2023.org for more information.
Well, is leaves changing color and going to watch that a thing where you are, Brenda?
Not as much as in the East Coast in the US. We do have, I think, some changes, but mostly it's just no leaves anymore. I don't like that. Winter with all the leafless trees, it's sad.
Oh, interesting. So,
Yeah.
I'm a huge fan of fall. It's my favorite season. Because you can wear pants and a short sleeve shirt with a jacket, and you can then take a jacket off. So it's like great for being thermoregulated. It has all of my favorite holidays, and it has Halloween and Thanksgiving and Jewish New Year and all of that. And fall food is on point. You know, it's warm enough you can still roast stuff outside sometimes, but cool enough you can do something inside. It's not just wet and dark and gross.
You're so optimistic. I just see the world more like that more often because all I think about fall and I think rain. I am in Amsterdam after all. It rains a lot.
You seem to be very a fan of summer and all the time off that you folks in Europe all get to seem to have all the time.
Yeah, the good life, you know. But I do think so. I in my hometown, down south. So fall starts in March and it goes into July, June, and actually I do like fall there because we do have nice tree color changes. It's actually pretty cool because you have this tree that goes really furiously red around May, and then you see it on the mountains, and it's like this strip of a red because it just like it grows between a specific height, specific altitude. So you look at the mountains, and there's just the green on the bottom, and then just one strip of red on top. That's kind of nice. So I guess there are pretty things in fall sometimes.
Now, do you like pumpkin spice lattes or not?
No.
Same.
No.
But I love pumpkin pie.
I don't think I've ever had a proper pumpkin pie. Jason is looking at me with like his eyes are just.
We have to fix that bit of lacking Americana.
Yeah. I think I like pumpkin and I like pie, so I think I would really like a pumpkin pie if I tried it. So
the spices and stuff, it's great. It's cinnamon.
Yeah.
Egg. It's all good.
Well, maybe you can bring one to South Africa. What about that? Can you bring one in the plane? How long do they stay?
I probably have a refrigerator on the seat I have. So,
Mr. First Class.
Mister, I have an arthritic hip and have to be able to function when I get there. Sure.
We all know you're old. Oh, arthritis. That's a good segue to my first paper.
Oh, please tell me how I cannot have to have a hip replacement before I'm 50.
Oh, gosh. Well, that I cannot promise. But now that we're already starting, let's just go for it. It is no secret that I am a big fan of regulatory T cells. But I think I will accept accept the fact that there are other cells that also, kind of dive into regulation. So although Tregs obviously do the heavy lifting here, there's many mechanisms that keep our body in homeostasis and when it comes to our immune system. And I think maybe one of the things that is wrong with you is your B cells are gone bad. So this paper I'm going to just talk about is called Antigen Presenting Autoreactive B Cells Activate Regulatory T Cells and Suppress Autoimmune Arthritis in Mice. And first author, Mike Aoun or Mike Aoun, not sure how to pronounce him, I'm really sorry, from the lab of Richard Holmdahl at the Karolinska Institute in Sweden. And I think this was really cool because in this paper, they discuss a population of B cells that are clearly autoreactive, but they're actually necessary or kind of an important regulatory element to prevent recognition of their antigen by the rest of the immune system. So what they do is they use a model. They study rheumatoid arthritis, and they have a mouse model in which they have this immunization with type 2 collagen, which can induce rheumatoid arthritis in mouse models. And they study, so they have a antigen, a model antigen derived from collagen that they use to study, antigen-specific responses. And so they use this kind of novel way to pick up B cells that are reactive against this antigenic epitope in the collagen. So it's called the C1 epitope. And they do this in kind of regular non-sick mice. And so they show that indeed in regular mice, in the spleen, in the bone marrow, there are, insignificant amounts of B cells that are reactive against this clearly self protein that is collagen. But despite them being there, they are not really mediating any autoimmunity. So the idea is that B cells are, in principle, during development, they're negatively selected, and B cells that have high reactivity against self-antigens are eliminated from the repertoire. But in the case of maybe affinities that are not so high, B cells, we know that B cells can escape this regulation. And then what happens to them? So they use this model to study what happens on a B cell that is clearly self-reactive, but it's not mediated autoimmunity. And they showed that it can actually have a regulatory role in the overall immune response. So what they find is that these B cells that I call C1b cells, they're found in not only in mice, but also equivalent ones are found in humans and human PBMCs. And they are indeed autoreactive. So if you actually force the reactivity, they will activate, for example, T cells that are reactive against collagen. They are really B cells that are recognizing an antigen that is self. However, if what they show in this publication that when they have a tendency to, so they in vivo in the mouse, these B cells are actually activating specifically regulatory T cells that are specific for this antigen. And in this way, they are not mediating autoimmunity, but rather the opposite. And they show that they do this. So some of the characteristic markers of these cells is CCR7 and CD72. CCR7 is a chemokine receptor that is basically homes cells to the lymph node. So this would give you the idea that these B cells go to the lymph node where they interact with T cells and with other cells. But CD72 which is an inhibitory lectin that is expressed in naive B cells. And this molecule and this interacts with certain, so they don't show exactly what it interacts with, but there's known ligands for this molecule on Tregs, and it mediates activation of presumably thymus-derived Tregs that are also recognizing the self-antigen. This also interestingly, this suppression mediated by these B cells is not dependent on IL-10, which is another known regulatory phenotype of certain B cell population. But this in this particular study, they seem to depend on the direct contact of these CD72 positive B cells with regulatory T cells of that are recognizing the antigen in question. So I think was very interesting. So they also show some human data. They also suggest that this is something that happens in a human, can also apply to the human situation, and that this could be a kind of a novel way in which we understand kind of tolerance or regulation of B cell responses by B cells that are, demonstrably autoreactive.
So you're saying B cells don't just make antibodies?
No, there's so much more to B cells.
That's the unsung theme of 23 that we've been kind of hitting on every so often. We had that at the AAI meeting and a few other guests on about this whole hidden world of B cells. But the fact that they're telling your Tregs what to do, does that make them more important than Tregs, Brenda?
Is what kind of loaded question is that? I mean, in the end, they're acting through, getting our friends, the Tregs, into, updated, and they are. So their main thing is they're acquiring this antigen and they're presenting it to Tregs and they're activating specifically Tregs. But yeah, I think it is important to give B cells. I think they're getting the day. What's the word? Under the sun, and I think yes, they're staying core. Well, a little bit more, less dramatic. But it's good. I mean, it is true that B cells are versatile actors in our immune system, and they're not only making antibodies. There's much more.
But you're also sounds like you're saying as a postdoc that the postdoc and then does more work than the PI, which you got to get the work done regardless of who's telling you where to go. So the Treg's got to do the work, even if the B cell is giving you some direction.
Well, yeah. I mean, is the B cell telling the Treg what to do? I think it's more, he's just sharing intelligentsia about, the environment, and telling the Treg will do what it has to do. The only element telling the Treg what to do, it's its own TCR. So it's its own internal drive to recognize antigen and suppress the immune system. So it's all intrinsic.
Just like a postdoc ready to have their own lab.
Exactly. I like that. That's more poetic. Thank you. So what do you have? So what's your story?
B cells and antigens and life or death and the meaning of all things. My article is in Immunity. It is antigen receptor signaling and cell death resistance controls intestinal humoral response zonation. First author is Fiona Razzo. Last author is Andrea Riboldi. Coming out of the Department of Pathology at the University of Massachusetts, Chan Medical School. So this came out, publication date's October 10th, but out now, obviously, since science publishes into the future. So that being said, this is an interesting article. As a reminder to people, B cell receptors are based on immunoglobulins. And so this is looking at the importance of IgA B cell receptor and what that does to those cells and other cells and the Peyer's patches. And they find that at a super high level, this is really about intestinal biology and really matters there and matters less elsewhere. So first off, IgA positive B cells dominate germinal cell reactions and are preferentially selected for in the memory compartment of Peyer's patches in particular. And this also is a little bit true elsewhere, but it really stands out in the germinal centers in Peyer's patches. And furthermore, if you get rid of it through knockout, and by the way, they have more mice in here. I think I need a chart. They have chimeras of knockouts, and then they have this whole system at one point where apparently if you have a heterozygote because of allele selection that happens at basically early development, if you have a set of heterozygote mouse, you're either going to have in that heterozygote, every cell will either be plus or minus, because only one gene is ever used. for the alleles for the B cells. So a heterozygote with one of each will naturally make a chimera, which I didn't know. And that is really interesting. So you get some really crazy genetics in terms of what they did. But they show IgA is required for homing the intestinal lamina propria. Other things like integrins are also important, but without the IgA, these cells do not home to the gut, generally speaking. So germinal center and homing. Then they go in and really start establishing through a combination of flow and staining that they really lead quite a bit of the process. So how is this working? They show that they look at the dark zone and the light zone and demonstrate that the fast mediated this isn't a dark zone process, but the fast mediated cell death process that occurs in the light zone. If you have a bunch of IgA B cell receptor, that turns that off. And that is the dominant mechanism. So you're having this positive selection for IgA positive cells in lamina propria. A, they get there and need IgA to get there. And B, they're relatively non-specific, right? So these IgAs cover many bacteria, not a single bacterial moiety on a specific strain, right? So they're a little bit more panoptic. They also show that it's the cell death in the light zone. So having this receptor activation keeps them alive there. And there's a little bit of a T helper component and that enhances it, but it's predominantly this cell death mediated thing. And the resistance to cell death, and they use like FASL negative, knockout systems, then makes it so that IgA cells are not dominant in the gut. So if you get rid of FASL mediated death for selecting the germinal centers, you no longer see a gut preference for IgA positive B cell, IgA positive B cells in terms of B cell receptor, which is really interesting. So it's saying it's really a key component of the immune selection process is this IgA in the gut. And then they show that the IgA mediates stronger B cell receptor signaling. And so especially in the gut centers, like the IgA positive cells have a much more robust response to pathogens, hemolyths, worms, pick something that matters in this system. It's the main driver of the effects that you would expect with IgA and humoral immunity in the intestine. So there you go. They really were trying to figure out the role of the receptor and where it comes from. The other thing they do is they do some kinetics here that I'm not getting into. It gets really complicated, but they show that it has to exist and then it moves to the gut and then it has the selection in the intestine. So they trace it all the way through.
Okay, so if I understand correctly, what is doing explaining why? So why would a B cell that makes IgA go to the gut? Because then they get this environment that is
right. So they're everywhere to begin with in the marrow. There's some even population everywhere, but they preferentially home to the intestine. If you are an IgA B cell, there's active selection based on this fast cell mediated process to kill everything else, or you're resistant to death if you have IgA, essentially.
Nice. I mean, that makes sense because you want most of your B cell space to be taken by B cells that make IgA that are necessary to keep the IgA that is needed in the gut, I guess.
Exactly. So this is basically showing how we end up with the known IgA predominance in the intestine.
Nice. Okay. I don't think of IgA too much. I'll have to admit. I like them. They're so good on my gut. Keep those bacteria in check. Okay. Well, we're gonna stay in the colon in the gut, because my story is again about another subset of cells that are trying to be regulatory. They do a regulatory job and they're good at it. So I think they deserve to be highlighted. So in this case, I'm gonna, with that on that lines and keeping my theme from last episode on gamma delta T cells, let's dive on gamma delta T cells and gut homeostasis. I think you're gonna like this story. The paper is called Conserved Gamma Delta T cell selection by BTNL proteins limits progression of human inflammatory bowel disease. First authors Robin Dart and Eva Zlatareva from the lab of Adrian Hayday at King's College London and the Francis Crick Institute in London. And in this study, they look into. I mean, they are aiming at understanding how different subsets of gamma delta T cells interact with different elements, because we know there is certain degree of diversity. Gamma delta T cells, although they have a kind of a limited repertoire, they do have some kind of specialization, and I think a lot is known, and even for people like me that are not in the gamma delta field, especially when it comes to the delta chains, the delta 1s, the delta 2s, and delta 3s, and the delta 2s are more common in more abundant in blood, and delta 1s and 3 are more common in like tumors, things like that. But then there is also some stratification when it comes to the gamma chain. And this is what they show in this publication, is that they look into the function of gamma delta T cells that are kind of differentially found between healthy gut and gut with IBD or with Crohn's disease or some kind of inflammation. And in this paper, they have samples from multiple donors, including healthy patients and people with inflammatory disease in their bowels. And they show that there's a particular subset of gamma delta T cells, which is the ones containing the V, so the gamma delta, so the gamma B4 chain, that do seem to, their absence seems to correlate a lot with the inflammation and the existence of an inflammatory disease. So these gamma 4 T cells are mostly recognizing two particular BTNL proteins. BTNL standing for butyrophilin-like proteins, which are the known ligands of gamma delta cells, and their distribution and their expression kind of varies across different tissues. And this delta 4, so it's gamma 4 T cells, they recognize these BTNL3 positive BTNL3 and BTNL ligands on the surface of tissues, particularly. And when they look, so they do a whole kind of characterization of the cells in these tissues, and sometimes it's not that easy, because oftentimes for measuring via FACS, there are no FACS antibodies against every single chain. So sometimes the absence of a chain kind of gives you the idea that the other chain must be there. So a lot of the data is also depends on transcriptomic analyses and things like that. So I have to say, it's a lot of things they measure. But basically what they say is that there is this population that is mostly expressing delta 1, and these are that they detect mostly the transcripts of gamma 4, and these are present mostly in healthy tissues, but not so much on inflamed tissues. And moreover, this participant population in question also expressed CD103, which is an integrin that is also very more often associated in T cells with tissue residency, also in alpha beta T cells, and this also gives the idea that these gamma delta T cells are really tissue resident. So what they I guess basically what they show is that the absence of this particular subset is correlated with increase with inflammatory disease, and that even there's they I think what's the most interesting part is they look into the expression of the ligands of these gamma 4 gamma delta T cells, as I mentioned, BTNL3 and BTNL8. And there are some polymorphisms that reduce the expression of these ligands, and these are associated in Crohn's disease. They're associated with increased inflammation, and also to some extent in IBD. So they also seem to kind of find in humans this correlation in the data from humans. So I guess that basically they suggest that this reduction in this distinct population of CD103 positive, V gamma 4 positive cells are associated with increased responses by the other, because what they show is that this particular population is reactive, so it does not react as strongly to their ligands as other gamma delta T cells, and that this is a way by which they're kind of dampening immune responses. So they have a rather suppressive phenotype compared to other gamma delta T cells. And so that's why they suggest that the absence of this population might actually have a detrimental effect, because then there's this immune modulation that is gone from the gut. So gamma delta T cells are also part of your gut. So how are your gamma delta T cells looking, Jason, you think?
Well, they're at least back enough with or the integrin inhibitors I have on board prevent them from leaving. We're not really sure. But it sounds like, do they know what causes them to go away? Like you have the correlation, but do they know the cause of the...
So I think in the case of the people with this polymorphisms, so basically because this mutant version of the BTNLs, they are, so I think it's, if I understand correctly, I think it's BTNL3 that is mutated, and also then the whole complex is expressed less. So the fact that they have less of these receptors being expressed, then that means that this particular subset of gamma delta T cells are receiving less stimulation, therefore either homing less or proliferating less, or like doing less of their suppressive function. So at least that is the situation when you already have some kind of baseline reduction of these BTNL proteins. But I don't think, like in regular people that have normal ligands, I don't know what would be the kind of the triggering thing to remove these cells.
Interesting. More to come then.
Yeah, but they do see that in both, so in Crohn's disease, they do see a reduction on the amount of these cells in people.
Very interesting. All right. Well, we're going to keep it to the gut. What a day. I love it. 3 out of 4 papers in the intestine. This doctor approves. All right. This is in Science. It is called Neuromedin-U programs eosinophils to promote mucosal immunity of the small intestine. So this is super interesting. It is first author Yu Li, last author Heping Zhu. They also look at pathogens, and that's a correction. I was switching up which pathogens. The last paper was looking at salmonella and other pathogens. This one looks at hemolyths for IgE. So we're class switching, Brenda. We're switching from IgA to IgE, and but we're still in the gut. So eosinophils, right, really important for type 2 immunity against things like parasites. A signaling in the small intestine that regulates it's pretty unknown. There's a protein called neuromedin-U, NMU, and a receptor, NMU-R1. Receptor 1, that's its receptor. They did a bunch of single cell and bulk analysis to start with to generate their hypothesis, and did some fate mapping, although who knows if that's what they really did first or not. That is what they presented in terms of order. They demonstrate that this signaling, the receptor expression in eosinophils, was programmed by the local microenvironment and enhanced the inflammatory response. So what do I mean by that? Essentially, and this paper is actually Very interesting, but also super hard to read. It has like 2 or 3 figures in it, or maybe 4, and like 15 supplemental figures with like 20 or 30 panels a pop. So it's actually kind of hard to follow the narrative if you're trying to look at the top line science, like just in the article itself without the supplemental. I think that's because they fit it in as the shorter form communication style. And so that then pushed a lot of it to supplemental. But just if you're reading through it, download the supplemental, folks. So they identify that eosinophils are really in the intestine, have this gene as compared to eosinophils necessarily elsewhere, which is kind of unique. So other eosinophils don't necessarily have it, but in the small intestine, if you're there, you have to have this. And then they created some lineage tracers using a tomato cassette and the appropriate Cree. And they demonstrate that and I want to get this right here. They persist in an active degranulation state. So they seem to be more active. They have less vesicles they're holding on to, and they're constantly in a degranulation form. Now, so that was their big first thing. So, hey, they're really important in the intestine. They constantly seem to be degranulating. Then they demonstrate that the neurons in the gut are responsible for the effects you see. So they demonstrate that through fate mapping that eosinophils will go to the intestine, start upregulating the receptor, and that then the ligand is coming from gut-specific neurons that generate their functionality to generate the signaling, which includes responses to hemolyths, right, to worms and all the other things you expect, and IL-25 related signaling. But if you look at responses to DSS, it doesn't matter. So this is very specific to what you think of as type 2 immunity, generally speaking, right? And they do this through a combination of lineage tracing experiments and chimeras as well, using the CD45.1 versus CD45.2 trick. And then they demonstrate that the presence of these eosinophils unshockingly leads to mucosal immunity, but that also regulates goblet cell differentiation. So if you get rid of them, you don't have as much goblet cell differentiation, and you also have decreased immunity. And they actually can demonstrate this in organoid culture and do some small intestinal organoid culture and knock out, take eosinophils with and without and put them in, and then see goblet cell formation, which is pretty cool to see. They also demonstrate that if you get rid of the gene, you get rid of the degranulation phenotype. You still have eosinophils. You just don't have the special eosinophil that we need in the gut to do its thing. And so it doesn't degranulate as much and generate that type of immunity. And that it is very specific that this is not super dependent on, so in a RAG knockout, it doesn't have an effect. But if you also get rid of IL-2, you start seeing a shift. Right. So just base RAG, which you would expect, right? Because what is, what's the adaptive immune system doing here at baseline? But if you start knocking out other stuff, then you get an effect. So this is a paper, if you are into eosinophils in the gut or looking at pathogen tolerance and parasitology in the intestine, it's important to know that there is a very key marker of the intestines that are doing this. There's this NMUR1 receptor and the signaling by the intestinal microenvironment through the neurons there to release the factor to generate this functional effect. Oh, and they also do things like DREAD receptors, which are those designer receptors exclusively activated by designer drugs. Thank you, Brian Roth, again, for that acronym. It is kind of cool. And they use that to drive the system on and off. So they can actually show, because they know how the receptor works as a GPCR, so they can pseudo turn it on and off through a DREAD and show that they can recapitulate the effects.
I guess I'm always fascinated by the studies that show this close collaboration between neurons and the immune system, and it's always cool to see.
I think, therefore I am, Brenda.
Yeah, I mean, it makes me think all the new age is like, well, we could meditate our way out of eosinophilia, I guess.
We're going to sign you up for a trial for that. But before we sign you up for that trial, we're also going to be speaking with Dr. Alice Long at the Benaroya Research Institute in just a few minutes here. But before we even get to that, there's one more thing, which is Stem Cell Technologies would like to introduce you to Immune Regulation News. It's a free weekly newsletter brought to you by Stem Cell Science News Program, covering research on the regulation, suppression, and modulation of the immune system. Immune Regulation News keeps readers current with the latest news, research, policy, events, and jobs relevant to the immunology community. Subscribe for free at immuneregulationnews.com. In other words, if you can't get enough of Tregs, T cells, and immune regulation, we got your answer.
Well, welcome everyone to the second part of the episode. Today we have with us Dr. Alice Long. She is associate member and principal investigator at the Benaroya Research Institute at Virginia Mason. And she's going to be talking to us about her work on understanding immune regulation on topic with our roundup today. And I guess also about her research on type 1 diabetes and what are the mechanisms that we can take in order to treat this disease. Dr. Long, thank you so much for joining us. By the way, this is the second time. You are our first repeat guest in the podcast. You joined us when we were having our AAI meeting special in 2022. So we're so happy that you're back.
Well, great. Thanks for asking me again. I'm actually honored. I didn't realize I was at the first repeat. It was a lot of fun the first time around.
Thank you for being here.
Yeah, of course.
All right, Brenda, you've gone first the last few times, every time, because it's been T cell. It's been very, after the microbiome mucosal immunity round, it's been T cells. I'll fire the first one off, but then I'll probably hand it back to your regulatory arms. So T cell autoimmunity, you focus on type 1 diabetes. Every autoimmune disease is different. So I'll put that out there as just a statement to start things. How T cell driven is type 1 diabetes as an autoimmune process?
Yeah, that's a great question. And I definitely agree that there are differences between autoimmune diseases, but there are also some similarities. There's tolerance is broken, so there's some commonality there. So I would argue that type 1 is more of one of the T cell focused autoimmune diseases. It has a very strong HLA association, which really does argue for CD4 T cells and the HLA interaction. And therapies that target T cells have been successful in type 1 diabetes. So in a prevention trial treating with teplizumab, which is a non-blocking, non-depleting anti-CD3 antibody, you can prevent and delay onset of type 1 diabetes with this drug. And there are other anti-CD3 targeting drugs, and there are other drugs that target T cells that can delay progression.
I guess you got to pick my attention when you discuss that there's some HLA restrictions that are associated with type 1 diabetes. Just because I like those kind of details. What are we talking about? Why? What is the mechanism behind that?
Oh, wow. That is a hard question. And unfortunately, that's something we've known.
As far as we know.
Yeah, no, but really, we've known about HLA associations for the autoimmune diseases for a really long time. And we're still struggling to get at the molecular mechanisms. So there's thoughts that this may be due to antigen processing and presentation, other things that might be due to levels of expression, but the HLA locus is complex. And so these just might be associations that, and chromosome structure is complex. And so it could be 3D and looping for the DNA also. So I guess we don't know.
All right, that was a concise answer. I guess that maybe when I think about it, I'm like, well, is it because some HLA subtypes have a higher, like this, they are more likely to accept a peptide derived from, I don't know, an islet cell or something? Is it along those lines? It's just because there's certain peptides that are very immunogenic and they're more likely to be presented by specific HLA molecules and not by others? And in that case, wouldn't we expect that to also be playing a part in establishing regulation or regulatory T cells in the thymus or negative selection in the thymus? I don't know. Am I along the right lines, or is it something
Definitely.
completely different that we think?
Yeah. So I think the tricky part is that's definitely part of the equation. And the association with air in the thymus with thymic selection is definitely an indication, along with the HLA locuses that are associated with type 1 diabetes. And the mouse model, NOD mouse model, definitely there's a link to how antigen is presented in HLA. In the human disease, not everybody has to have those HLA loci to have type 1 diabetes. So it's a little more complex, but definitely there's something to that component to it. Yeah. How that controls regulation and what we've seen in terms of T cell exhaustion and that being associated with response to therapies, we really don't know at all.
So then it essentially the working hypothesis is that certain HLAs are just better, or actually worse, but in this case, better at picking up specific antigens, which we know. And in this case, those antigens end up being self-antigens, which leads to a problem. Instead of, killing a pathogen, you get yourself in the crossfire. But it's based on that HLA's predilection to anthropomorphize things a little bit. But its binding is favorably bad for us.
Yeah, I think that's a working hypothesis. But honestly, the peptide could be presented and it primes the T cell and to go in one direction or another also. So kind of work both sides of that equation.
There's also different types of type 1 diabetes. No, you have those that are that kind of subset of disease that is more likely to present in young people, and then other that is more predominant or slightly different presentation in older people.
So type 1 diabetes is autoimmune by definition. We don't really know that there's a difference between childhood and adult type 1 diabetes. Yes, children often progress faster, but whether that's just a rate of disease or the actual mechanism is different, I don't think we know. And as a community, I think we're still trying to figure out whether there are types of type 1 diabetes, like an endotype, kind of it's putting a word to a concept that I'm not quite sure we've proven yet in the field. Definitely people don't all respond to the same therapy. So put it that way, that something's different about their disease. Now, whether that's where they are in the progression of the disease or the immunology is different. There's more involvement of the beta cell and disease pathology in some people and immune cells and others. I don't think it's known yet.
So then therapeutically, you said, non-depleting, which is good, right? Since you're not going to get rid of all the T cells, seems to work. And if it has such a strong autoimmune component, why do you think it's been so hard with, let's say, hey, I am a kid or an adult, I'm showing some type 1 diabetic signs, like some rising A1C, but I'm not so far gone that I have zero beta cells left, because once you're at zero beta cells, you obviously need a beta cell transplant. That's the other podcast. That's a stem cell podcast. But if you got some left, but your immune system's starting to behave badly, my understanding is that therapeutically, we haven't been able to really stem the tide in a way that it's a standard protocol now in patient care. What's the barrier to that?
Great question. And you could probably frame that question with any autoimmune disease. Type 1 is actually pretty exciting. So in the sense that we have autoantibodies to islet antigens that we can detect early in the disease process. And so you're clinically diagnosed with type 1 diabetes when your pancreas can't make enough insulin. And so you have to use insulin as a replacement therapy. And so that's what you would do instead of a transplant until those are available, when your pancreas really can't make enough insulin. So we have actually shown that treating with anti-CD3, when you have autoantibodies and some level of dysglycemia or your beta cell is sick, but you don't yet need insulin, we can delay that time to needing insulin. So it's actually one of the autoimmune diseases that we can come in early and actually prevent full on clinical presentation. And that's really outstanding and unusual, because most autoimmune diseases, we wait for the clinical manifestation and then we try to treat the disease. So I'd argue that we've done a little bit in that space, but there's more to be done.
So nowadays, what are the so you mentioned anti-CD3 treatment. Is that the best we have in terms of modulating the immune system to prevent the kind of the full onset of diabetes, or to treat already something more established?
In terms of immune modulation, that is the only drug approved to date for type 1 diabetes, which makes diabetes unusual from other autoimmune diseases too, because other autoimmune diseases have a number of other options they can try for treatment. And because of the availability of insulin as a replacement therapy, we have a higher bar to reach before we can go for immune therapies. But when we've shown that we can prevent the onset of type 1 diabetes, that really is good rationale.
Yeah, which would explain things like steroids and stuff being off the table because of their side effects and everything else. And
they do work. They did try those, but yes, they're off the table because of the side effects. Yeah.
well, they also raise your blood insulin levels. So the treatment makes the problem worse acutely too, which isn't great. So then, how do you think the lessons from type 1 diabetes work can be applied to autoimmunity more broadly? To do the reverse of my first question.
Yeah, so I actually think the concept of, and others have shown, so Owen McKinney's done some nice work also to show that T cell exhaustion likely is involved in other autoimmune diseases in a beneficial way. To this date, this has all been association. So I do want to put that out there. We're trying to figure out what might be going on and the mechanisms underlying those. But in terms of T cell exhaustion, I think there's a good argument that it is involved in at least more than one autoimmune disease. Maybe not all, but at least more than one.
So when you talk about T cell exhaustion, so you just mentioned that we think it might have actually a beneficial correlation. How should we think about, because often we're thinking of T cell exhaustion in the context of either cancer or maybe chronic viral infection. So I don't think a lot about T cell exhaustion in the concept of autoimmunity. So do we think, could that be some kind of adaptive response? Like, well, it's actually a good thing? Or do you think it's just a side effect of the exhaustion, the traditional exhaustion, or how do we understand exhaustion in the context of autoimmunity, and particularly, I guess, CD8 cells?
Yeah. So I think the question is kind of two parts, in the sense there's exhaustion in the context of therapy, and that's kind of what is the drug doing, and I think that's kind of signal one without signal two or three, where you do get exhaustion, and that's associated with better response. But we have seen increased levels of exhaustion in slow progressors and islet-specific cells, and so whether that's taking up space for other cells that are lytic, whether that is tissue repair. So if you think about it in the context of a chronic viral infection, those cells aren't capable of completely getting rid of the virus, but they are capable of getting rid of enough and getting rid of the damaged cells so you keep the tissue intact. And so maybe the pancreas is already starting to be sick and stressed, and you're getting rid of those few sick and stressed cells and leaving the others to do the job. So that would be one argument, neither of which we have solid proof for yet, but those are hypotheses that are being pursued.
But are these exhausted cells for sure kind of antigen specific?
Yeah, so we have shown in the natural history of disease that people that have slow progression do have islet-specific increased levels of exhausted cells. And the therapy they are more global. The therapy targets CD3 cells. Within those, yes, the islet-specific cells do also increase in exhaustion.
So it sounds like then, T cell exhaustion is usually air quotes bad, right? But it sounds like in this case they're air quotes good. Does that mean that one of the therapeutic angles that we're going after now, we being the general sense here, is exhausting T cells deliberately?
Yeah, I would argue that. Yeah. And kind of another piece of evidence to that maybe being beneficial is the checkpoint blockade for cancer. When that occurs, you get autoimmunity as a adverse event. And so that's all about getting rid of exhaustion. So if we can figure out how to do the inverse, that would be great.
Have there been documented cases of people having I don't want to say getting diabetes after cancer treatment, but is that something that actually happens, that you exacerbate autoimmune diseases to a point that you maybe cure your cancer, but you get something else?
That's exactly what happens with about, I think it's about a quarter of subjects treated with.
So it's not temporary. Because I know that you have, a cytokine release syndrome, things like that. Most people are maybe treated with tocilizumab or things, but you're talking about long-term autoimmune disorders that occur after checkpoint inhibition.
Mm-hmm. Yep.
I was not aware that the number was so high, like long-term complications.
Isn't that usually immune checkpoint colitis?
There's a lot of colitis. Yeah, it's a whole range of autoimmune diseases.
Yeah, how much type 1 diabetes comes after checkpoint blockade for?
I don't know the exact numbers for that, but it's not super rare, or at least endocrine diseases in general.
That's a side effect I wasn't thinking about for checkpoint. Well, I mean, of course, it makes complete sense when you spell it out like that.
But it's treatable, so
yeah.
I think of it from the patient's perspective. They're looking for some extra time. Yeah.
And so we're talking about T cells, of course. Bad T cells, islet specific, or other, detrimental T cells activating in the pancreas. What about Tregs? Are you at all looking into, in your research or in your interest, are you looking at what can we do to maybe increment or intervene in the context of Tregs and type 1 diabetes?
Yeah. So in my research, I've been focusing more about kind of understanding the molecular mechanisms of exhaustion, what drives it, how those cells functioning. But I'm also involved in a lot of clinical trials in the type 1 diabetes space. And certainly there's still remaining interest in figuring out how to augment regulatory T cells and think about when in the disease progression that might actually work best. Is Treg regulation something that's lost early, where we need to kind of think about treating early in the disease stages with Tregs, and maybe later in the disease that's a little too late, and that's when you need to think about exhaustion.
Makes sense.
So jumping back to exhaustion, which I don't guess you don't jump to exhaustion, but you slowly roll into it. What are the main molecular mechanisms that you've been able to dig up? Like what, you're going to go, we talked about, it'd be great to drug it and cause exhaustion. Well, what causes exhaustion? Besides sleep?
Yeah. So we don't have any answers yet, but definitely T cell receptor signaling that is sustained instead of episodic. And so how that occurs, when that occurs, and how you can make that occur naturally is a challenge. We do interestingly see increased levels of exhaustion with some islet antigens and not others. So we're trying to frame some of our questions around that to understand why some individuals may have different levels of exhaustion based on specificity.
So maybe some TCRs are stronger and they're over. I think this is what we see also in the cancer situation, that there's certainly different tiers of TCR affinities, and those that have very high affinity are often very, they can be very efficacious. but then they're usually the first ones to get exhausted, and then you end up with a different kind of response in the long term. So I guess, is it also some kind of quality of different TCRs that you can measure?
Yeah, I wish we could. That was one of our first obvious questions, and that didn't seem to pan out quite right. And I don't know if we weren't looking in a precise enough manner or had the right tools, or that just wasn't the answer. But yeah, we don't yet know. We're still posing those questions. And the challenge in the humans is kind of getting the right comparisons to do it. And what people do you compare to the others, or what specificities within an individual do you compare to each other? So
Yeah, I guess between the diverse, I mean, HLAs are very diverse. So if that is a driving factor, then you're going to have a lot of diversity amongst humans. And I guess that also maybe mouse models, well, it's always a limitation, right? Using mouse models to study a human disease, at some point it gets, especially if you're talking about T cell repertoire, things like that are so human, then must be.
I think that mouse models would be great. It's just not something that I've developed as much in my own lab, and so something that I would do in collaboration with somebody else.
So it looks like there's a lot still to learn about immunity of type 1 diabetes. And let's hope that we can find something, or that the guys at the stem cell side just make some artificial pancreas, and then we can all be, it all can be solved. But of course, a huge issue, especially nowadays with this kind of diseases being ever more prevalent. So the need is obviously there.
And the field is not exhausted yet. Sorry, I had to.
Nice. Love it. Excellent pun. So to finish on this high, on this how you say, high note. So I like to ask our guests kind of lateral question at the end of our show, just so that the listeners can get to know a little bit more about them. So we would like to ask you, Alice. So is there a hobby that you always wanted to pursue and never had time to? And if not, if there's a hobby that really defines you that you would like to share with our listeners today?
Yeah, maybe I'll pick a hobby that I haven't had time to do yet, but would still be on the wish list. And that's to kind of see more of the underneath of the deep sea. I'd love to be on those little submersibles where you can look out of the glass and kind of just troll around down there and see what else is down there. It seems like a whole nother world.
Okay, but don't go see the Titanic. Just that's the one you should stay away from.
Oh, too soon.
All the creatures.
Too soon. I'm sorry.
Too soon.
Sorry.
Too soon.
That was not. That was a little bit unrequested. But that sounds so cool. Like Captain Nemo and the 20,000 Leagues Under the Sea situation. More like that. Let's go for that. For the epic. Have you ever done any diving of any type?
No, I like to swim and I snorkel, but I haven't even scubaed. So yeah, I'd be just doing it whole hog.
You do scuba dive, right, Jason?
I little, not very much. I haven't done a ton of that either.
Well, we had a couple of guests that were scuba divers enthusiasts. So maybe we should get you in touch, and then maybe you can just you can start. Maybe not in a submarine, but maybe it's just a little bit more deep. Or you can ask James Cameron for his submarine. That one will get you to the Titanic for sure. Well, it's been a pleasure talking to you today. Thank you so much for joining. And we wish you success. I hope that we get to see you in next AAI meeting. We couldn't see you this year, but let's see if we can run into each other next year. Jason, anything to add?
Nope. We'll leave it there. But I guess I was going to say AAI is not next to an ocean next year, so we could go scuba diving. But one time it will be.
Yeah, 2025. That's Hawaii.
There we go. All right, Scuba Hawaii 25. Done. Brenda knows where Hawaii is.
We're here for the long run. We're thinking ahead.
Multi-year view, always important.
All right. Thank you so much. And we'll stay in touch.
That sounds great. Thank you.
That brings us to the end of our show. Don't forget to subscribe to our newsletter at www.immunologypodcast.com to get the show notes, including an episode summary and links to all of the interview and roundup papers. You can also reach out to us on Twitter at @immunopodcast or via email at info@immunologypodcast.com with feedback or to suggest guests. See you next time.